let us look at proteins. So before we proceed, kindly like, share and subscribe to my channel. That will encourage me to post more and more videos, guys. So before we go to proteins, previously we were looking at amino acids. So amino acids refers to carboxylic acids that contain amino groups. So we describe amino acids as monomers of proteins. So amino acids refers to the monomers of proteins. We went ahead and looked at the structure of amino acid. So the basic structure of amino acids consists of a central carbon, a carboxyl group, an amino group, a hydrogen atom, and a side chain, which normally distinguishes one amino acid from another. So in the same lecture, we went ahead and looked at the classification of amino acids. So amino acids can be classified based on the structure of the side chain, on how they appear on the side chain. So they can be classified as amino acids with side chain containing alkyl groups, that is carbon and hydrogen atoms only. So we talked of glycine, alanine, valine, leucine, and isoleucine. Then we also went ahead and selected some group of amino acids that contain sulfur atoms only. So if you can remember cysteine, cysteine contains sulfur atoms and a sulfur atom on the side chain. Then we also looked at the amino acid whose side chain contain a cyclic compound. So those ones, we looked at tyrosine, we looked at phenylalanine, we looked at tryptophan. Then there are also amino acids whose side chain contain carboxyl groups. Those are acidic groups. So those are aspartate and glutamate. So their names end in ATE. Then there are amino acids whose side chain contain amino groups, and they normally yield hydroxyl groups on hydrolysis when they dissolve in water. So those ones are glutamine and aspargine. Then we went ahead and looked at another classification, a broad classification of amino acid that is based on the nutritional importance. So amino acids can be classified as essential and non-essential amino acids. So examples of essential amino acids, we talked of tryptophan, then threonine, histidine, valine, isoleucine, and so forth. So we mentioned 9 essential amino acids, then the others are non-essential. We looked at the difference between essential and non-essential amino acids. We say that essential amino acids are those amino acids that must be provided in the diet. So the body requires them for survival, while non-essential, they can be synthesized by the body and therefore they are not supposed to be, they are not important in the diet. That is it. So after that, we looked at the peptides. That's another lecture. We looked at the peptides, still on the amino acids. So we said that peptides consist of 2 or more amino acids, which are normally linked together by a peptide bond. So during the formation of a peptide, there is normally a loss of water molecule, and we looked at that. We looked at an illustration where we were combining one amino acid with another, and there is a loss of water molecule in the process of hydrolysis to form a peptide bond. We also drew the structure of glutathione in the same lecture. So in this lecture, let's look at the proteins. So proteins are macromolecules which hold the first rank in the bodies of living organisms. So these are polymers. They are polymers of amino acids. So they are made up of 2 or more amino acids. That's why they are called polymers of amino acids. They are macromolecules. They are large molecules. So what are the importances of proteins? So the first importance of protein is transport. And the first protein that we met even right from our childhood is hemoglobin. Hemoglobin refers to that protein molecule that contains that has got that is normally red in color, that is normally involved in the transport of respiratory gases. So the respiratory gases are essentially carbon dioxide and oxygen. So hemoglobin is normally involved in the transport of respiratory gases to respiratory site, and that's an example of proteins. Then another importance of protein is defense mechanisms. And those ones that are involved in defense are immunoglobulins, so the white blood cells. Then we have control of gene expressions. An example of such a proteins are histones. We are going to look at histones on the biochemistry of nucleic acids. That will be in the next lecture. So proteins also act as hormones, which normally control many biochemical events. So you know that hormones are biochemical messengers in the chemical reaction. So some proteins provide strength and elasticity to organs and vascular systems, e.g. collagens and elastins of bone matrix and ligaments. So let's look at the properties of proteins. So the first property is a charge. So the charge of a protein can be altered depending on the pH of the surrounding. We can change the pH of the surrounding solution if we want to alter the charge of a protein. So we looked at that one in the case of amino acids. So that is the same thing here. Then isoelectric point. So isoelectric point refers to that point where the charge of a protein is neutral. So at an isoelectric point, we have this is a point at which the protein exhibits a dipole moment where it has got both positive and negative charge. So the amount of positive charge and the amount of negative charge on a protein are equal, so they cancel each other. At an isoelectric point, the protein does not move in an electric field. Then we have higher molecular weight. So we've just said that these proteins are polymers of amino acids. So they contain more than one amino acid. And other than that, some proteins go ahead and consist of prosthetic groups. These are conjugated proteins. So we expect them to have higher molecular weight. And an example of such a protein is pyruvate dehydrogenase. We looked at the pyruvate dehydrogenase enzyme, that is in pyruvate decarboxylation. Then another property is they act as buffers. So buffers are salts of weak acids and bases which normally resist change in pH when an acid or an alkali is added to them. So hemoglobin accounts for 60% of buffering action within the erythrocytes, whereas proteins account for 20% of buffering action of blood. Then another property is hydrolysis. So we know that proteins are polymers of amino acids. So if it's a polymer, when it undergoes hydrolysis, we expect it to yield its components, the constituents. So on hydrolysis, amino acids proteins normally yield amino acids. So since we have various types of proteins, there are those ones that are conjugated proteins, and then we have simple proteins. So conjugated proteins are those proteins that consist of prosthetic groups. So these ones on hydrolysis, they yield amino acids plus the prosthetic groups. Those ones that are simple, they only yield amino acids. So what are conjugated proteins? They contain permanently associated chemical components in addition to an amino acid called prosthetic group. So we go ahead and look at the prosthetic groups. Examples of prosthetic groups, we have lipids. So lipoproteins contain lipids as prosthetic groups. Then we also have glycoproteins. Glyco means sugar. So glycoproteins contain sugar molecules in their groups. Then metalloproteins, they contain specific metals on hydrolysis. So conjugated proteins normally yield amino acids and prosthetic groups. So we go to classification of proteins. So proteins can be classified based on the composition or structure, then also based on the solubility and based on their shape. So based on their composition or structure, we can have simple amino acids and also conjugated amino acids. I have already explained that. Then we also have derived amino acids. These are amino acids that are derived from simple and conjugated amino acids. So these ones are synthesized. Then amino acid proteins can also be classified based on the solubility. So based on the solubility, we can classify them as albumins, globulins, glutenins, and histones. So albumins dissolve in water and salt solution, for example, plasma albumins and egg albumins. Then we have globulins. Globulins are sparingly soluble in water but soluble in salt solution. Then glutenins are soluble in dilute acids and alkalis. Then we have histones. Histones are soluble in water and dilute acids. We go to classification based on their shape. So we have globular proteins and fibrous proteins. So globular means the globe. So the polypeptide chain of their proteins are folded into compact spherical shape. For example, hemoglobin is a globular protein, Hb. Then fibrous, the polypeptide chain extend alongside one axis. So what are we saying? We are saying that proteins are polymers of amino acids. So we go to the importance of proteins. The first importance of proteins is transport of substances. Another one, we've said that they act as enzymes, defense mechanisms, they control gene expression, and some of them act as, they provide strength and elasticity to organs and vascular systems. So we go to properties of proteins. The properties of proteins involve number one, charge, isoelectric point, higher molecular weight, act as buffers. So as a charge, we've said that we can alter the charge of a protein by changing the charge of the surrounding, the pH of the surrounding. Then the property of isoelectric point, I have said that at an isoelectric point, the protein does not move in an electric field because it is electrically neutral. The number of negative and the number of positive charges are equal. So go back to the lecture on the lipids. Then another property is higher molecular weight. So proteins they are made up of amino acids, and some of them contain conjugated then prosthetic groups. So we expect them to have higher molecular weight. Then proteins also act as buffers. So I have described what buffers are on the lecture of amino acids. Then the last property is hydrolysis. So since proteins are compounds and a compound contain two or more elements, so on hydrolysis, proteins yield their components, which are amino acids, and also those ones that contain prosthetic groups, they yield amino acids and prosthetic groups. So kindly like, share, and subscribe to my channel. Have a good day.